Welcome back to the deep dive. Today we are shifting gears. We aren't just reading a textbook. We're effectively walking onto a crime scene. We are putting on our detective coats.
I like the sound of that. A molecular detective agency.
Exactly, because if you really think about it, biology is just this massive, ongoing, chaotic mystery. You have this incredible, complex system, the human body, and things are happening constantly.
All the time.
Muscles contract, thoughts fire, food gets digested, diseases strike. But to solve any of those mysteries, to understand, who doing it or how doing it, you can't just look at the whole body at once. It's just too big.
It's overwhelming. You have to zoom in.
You have to zoom way in. You have to look at the smallest players in the room. You have to interrogate the suspects.
All right.
And that is exactly what we are doing today. We are pulling the files on the most fundamental cast of characters in all of biology. We were talking about amino acids, peptides, and proteins.
The very alphabet of life.
Yeah.
I mean, it all starts here.
It really does. And to help us solve this case, we are synthesizing two absolute titans of the biochemistry literature. We've got Leninger's Principles of Biochemistry.
Which is going to give us the beautiful chemical logic, the rules of the road.
And we've got Harper's Illustrated Biochemistry.
And Harper's brings the clinical reality, the diseases, the poisons, the medicine. It's the what happens when it all goes wrong manual.
It's a powerful combination. I mean, think of it this way. Leninger tells you how the machine is supposed to be built, right? The physics that holds it all together.
Yeah, the blueprints.
And then Harper tells you what happens when the machine breaks down or, when someone throws a wrench in the gears.
A very, very tiny wrench.
A molecular wrench.
Yeah.
So let's set the scene. We are not just going to memorize a list of chemicals today. I want to treat these amino acids like suspects in a lineup. They have personalities, don't they?
Oh, they absolutely do. In biochemistry, structure is personality. It's everything. If you know what a molecule looks like, where its bumps are, where its charges are, you know exactly how it's going to behave.
And the stakes couldn't be higher. We're not talking about generic bricks in a wall that all look the same.
Not at all. These tiny molecules, they're neurotransmitters. They're the precursors to hormones. Some of them, if they show up in the wrong place, are actually deadly poisons.
And if you misidentify one, if you swap a single one of these characters for another in a protein chain, the entire system can just collapse.
That is not an exaggeration. We see that in genetic diseases all the time. One wrong character in the code, and the protein can't fold, or it clumps up, or it just loses its function entirely. A single distinct atom out of place can be the difference between health and a debilitating condition.
So our mission today is to move past that rote memorization. I don't want to just list names. I want to understand behavior. By the end of this deep dive, I want to be able to look at an amino acid and predict what it's going to do at a party. Is it going to hide in the corner? Is it going to start a fight? Is it going to hold hands with everyone?
That is the goal. If you understand the personality of the players, you can predict the outcome of the game. You can see the whole story unfold.
Okay, so let's start with the anatomy of a suspect. We need a standard template. If we pull an amino acid into the interrogation room, what the textbooks call an alpha amino acid, what are we actually looking at? What's the basic body plan?
We are looking at a central hub. In almost every case, we are dealing with a molecule that is centered around one specific carbon atom. We call it the alpha carbon. You can think of it as the torso of the suspect.
Okay, the torso, the central point. And attached to this torso, we've got 4 appendages.
Correct. And the interesting thing is that for every suspect in our main lineup, 3 of those appendages are effectively wearing the same uniform. They are, for all intents and purposes, identical.
Walk me through the uniform. What are the 3 standard parts?
Okay, so first you have a carboxyl group. Chemically, that's your acid component. It's a carbon with 2 oxygens.
The acid.
Got it. Then on the other side, you have an amino group. That's a nitrogen with hydrogens. That's your basic component.
Acid on one side, base on the other.
Exactly. And then usually tucked in the back, purely for balance, you have a simple hydrogen atom. So carboxyl group, amino group, hydrogen, that's the standard issue uniform. Every single one of the 20 standard amino acids has this setup.
So if everyone has those 3, what makes them different? Why isn't alanine the same as tryptophan?
It's the 4th appendage. The R group, the side chain. That is the variable. That's what defines the identity of the amino acid. It's like everyone is wearing the same gray jumpsuit, but they're holding a different tool in their hand.
Oh, I like that.
Yeah, one might be holding a fire extinguisher, another a magnet, another, a glob of grease. That tool is what determines what they can do and who they can interact with.
Okay, but before we get to the tools and the specific suspects, I want to unpack something that came up in the sources, specifically in Leninger. This concept of the Zwitterian. It sounds like a sci-fi villain.
It does, doesn't it? Attack of the Zeterians.
Yeah, exactly.
It comes from the German for hybrid ion. And honestly, it is absolutely fundamental to understanding how these things exist in your body. It sounds complex, but it's just describing the electrical state of the molecule.
Okay, so break it down. What makes it a hybrid?
Well, let's do a thought experiment. Imagine taking a single amino acid, let's say a generic one, and dropping it into a beaker of water. Now, imagine we have a dial that controls the pH of that water.
Okay.
If you turn the dial to a very low pH, so it's highly acidic, the solution is just swarming with protons. It's a proton mosh pit.
Protons everywhere.
In that environment, both the amino group and the carboxyl group are going to grab protons. They're greedy. The amino group becomes NH3 plus AO, and the carboxyl group becomes COOH. The whole molecule has a net positive charge.
Okay, so at low pH it's positive. Now I spin the dial the other way. High pH, very basic.
Now, the protons are scarce. The environment is desperate for them, so it strips them away. The carboxyl group loses its proton and becomes negative, COO. The amino group also loses its proton and becomes neutral, NH0. So now, the whole molecule is negatively charged.
But our bodies don't typically run at pH 1 or pH 11. We sit in the middle.
Exactly.
Yeah.
Physiological pH is around 7.4, and this is the Zwitterian zone. At this specific pH, something really interesting happens. The carboxyl group, which is a stronger acid, has already lost its proton, so it's negative COO.
Right.
But the amino group, which is a weaker acid, is still holding on to a proton, so it's positive NH3R. So you have a single molecule that has both a positive charge and a negative charge on the same structure at the same time.
So the net charge is 0.
The net charge is 0, yes. But the molecule is highly charged locally. It's a dipolar ion. And this matters immensely because it dictates solubility. Because they have these charges, they love water, they are soluble in polar solvents, they dissolve in your blood, they dissolve in the cytoplasm of your cells.
If they weren't Zwitterians, if they were just neutral and greasy, they wouldn't float around in our cells the way they do.
Exactly. They would crash out of solution, they'd clump up. Life as we know it relies on this electrical schizophrenia, to keep these fundamental building blocks mobile and available. They do not dissolve in nonpolar solvents like benzene. They stick to the water.
That's a key point. Now, there's another structural rule we have to hit before we meet the cast, the concept of handedness, chirality.
Right. So the alpha carbon, that central torso, is a chiral center. And what that means is it has 4 different things attached to it: the amino, the carboxyl, the hydrogen, and that R group tool.
And whenever you have a carbon with 4 different partners, you get mirror images.
You get mirror images, just like your left hand and your right hand. They look the same, thumb, fingers, palm. But you can't superimpose them on each other. You can't put a left-handed glove on a right hand. They are non-superimposable mirror images.
And the sources make a really big deal about this. Nature isn't ambivalent about hands. It is incredibly picky.
It is. It's one of the great mysteries. Proteins in the human body and in almost all biology on Earth are an exclusive club. They only allow L-amino acids.
L for left.
Essentially, yes. It's a chemical convention related to the structure of L-glyceraldehyde. But the takeaway for us is that our entire machinery, our enzymes, our receptors, are all built to work with the L isomer. It's a lock and key mechanism.
So if you try to jam a D-amino acid, the right-handed version, into an enzyme active site,
it just won't fit. It's the wrong shape. The geometry is all wrong.
But here is where Harper throws in a curveball. The sources mention that D-amino acids do exist in nature. We don't use them in our proteins.
That's right. And this is clinically really important. You find D-amino acids in bacterial cell walls. They use them as structural components. And you also find them in certain peptide antibiotics like bacitracin or gramacyride A.
Which is actually incredibly useful for medicine, isn't it?
Hugely useful. Because if bacteria use D-amino acids to build their walls, and humans do not, that gives us a perfect target. We can design drugs that specifically attack the D-amino acid structures or the enzymes that handle them.
It allows for selective toxicity. We can bomb the bacteria without hurting the human host because the handedness of our architecture is different.
Exactly. It distinguishes friend from foe at a fundamental geometric level.
And there was one other detail about chirality that it blew my mind. The alien connection, the Murchison meteorite.
Ah, yes. This is fascinating stuff. When we analyze meteorites that contain organic compounds, literally rocks falling from space, we find amino acids. But in the meteorites, we find a mix. We find both L and D isomers in roughly equal amounts.
Which suggests that abiotic chemistry happening out in the void of space without life involved doesn't care. It makes both hands.
Exactly. Extraterrestrial organic chemistry isn't picky, but life on Earth is. Somewhere, billions of years ago, when the first replicating molecules were forming, life chose the L path, and we've been stuck with it ever since.
That is a deep thought to keep in our back pocket. We are living in a left-handed biological world. Okay, let's get to the lineup. We have 20 standard suspects to identify, and the sources classify them based on their personality, specifically how they feel about water.
The polarity scale. This is really the most useful way to group them because it tells you where they're going to live in a protein. If you hate water, you're going to bury yourself deep inside the protein to get away from it. If you love water, you're going to want to be on the surface.
Let's start with group 1, the introverts, the nonpolar aliphatic group. Who are they?
Okay, so we have 7 members here: glycine, alanine, proline, valine, leucine, isoleucine, and methionine.
And their defining trait is that they are hydrophobic, they're greasy.
They fear water. But I want to clarify something here because it's a common misconception. We say they fear water, but in reality, it's more that water rejects them.
Rejects them. How so?
It's the hydrophobic effect. So water molecules, they love to form hydrogen bonds with each other. They create this very tight, very ordered network. When you introduce a non-polar, greasy molecule like leucine, it disrupts that network.
It gets in the way.
It gets in the way.
Yeah.
It forces the water to form this ordered cage around it, which is energetically expensive.
Oh.
It lowers the entropy. So to minimize that disruption, the water literally pushes these nonpolar molecules together. It crushes them into the center of the protein to minimize their surface area.
So the protein folds not just because the amino acids like each other, but because the water is forcing the introverts into the corner of the room to get them out of the way.
That's a perfect way to put it. That clustering is what stabilizes the core of a folded protein. It's the glue holding the center together.
Let's pick out a few distinct characters from this group. Start with glycine.
Glycine is the baby of the family. It has the smallest side chain possible. It's just a single hydrogen atom.
So it's tiny.
It's tiny. And it's the only one that is not chiral because its R group is a hydrogen. So it has 2 identical hydrogens attached to that central carbon.
What's the consequence of it being so small?
Because it's so small, glycine can fit in tight corners where no other amino acid can go. If a protein chain needs to make a really sharp turn, or if 2 backbones need to get really close, you will almost always find a glycine there. It provides flexibility.
It's the contortionist of the group.
Exactly.
Okay, what about proline? The sources call it an amino acid, which sounds different.
Proline is the rebel. It's totally unique because its side chain loops back around and connects to its own amino group on the backbone. It forms this rigid 5-membered ring structure.
So it's locked in place.
Extremely rigid. It's locked in place. While glycine is super flexible, proline is the opposite. It's stiff. It creates a kink in the chain. It's actually known as the helix breaker because it can't fit into the smooth spiral of an alpha helix. If you see a proline in a sequence, you know the protein structure is going to have a sharp kink or a rigid turn right there.
It dictates the architecture by limiting movement.
Precisely.
And what about methionine?
Methionine is one of the 2 sulfur-containing amino acids.
Oh.
But here, the sulfur is sandwiched between 2 carbons. We call that a thioether. It's kind of hidden away and non-reactive, so methionine remains nonpolar. It's famous for being the start signal for protein synthesis, but structurally, it's mostly just another greasy brick in the wall.
Okay, moving on to group 2, the show-offs, the aromatic group.
Phenylalanine, tyrosine, and tryptophan. These are the big boys. They have these big, bulky, flat ring structures in their side chains.
They're also relatively nonpolar, so they tend to be hydrophobic.
For the most part, yes. But they have a special talent, a chemical fingerprint.
Fingerprint. They absorb UV light.
They absorb UV light, specifically around 280 nanometers. Those ring structures are perfect for it.
And why does a lab scientist care about that?
Oh, it's one of the most useful tricks in the book. It is the standard way we measure protein concentration. If you have a test tube and you want to know if there's protein in it or how much, you put it in a spectrophotometer and you shine UV light at 280 nanometers through it.
And if the light gets absorbed, you know you have protein.
Exactly. Tryptophan is the real star here. It absorbs UV light about 4 or 5 times more efficiently than the others. If there's an absorption peak at 280, you can be pretty sure you have protein.
So tryptophan is the beacon.
It is. And we should also give a shout out to tyrosine here. Harper points out that tyrosine has a hydroxyl group, an OH, on its ring. Phenylalanine is just a plain hydrocarbon ring, but tyrosine has that little chemical handle.
That makes it a bit polar, right? It can interact with water.
Slightly more polar, yes. It can form hydrogen bonds, but more importantly, that OH group makes it reactive. It's a handle that enzymes can grab onto. It's a key site for chemical modification, especially phosphorylation, just like we're about to see.
Which is a perfect segue to group 3, the social connectors. Polar, uncharged.
Serine, threonine, cysteine, asparagine, and glutamine. These guys are hydrophilic. They love water because their side chains can form hydrogen bonds. You're going to find them on the surface of proteins interacting with the cellular environment.
And serine and threonine seem to have a special role regarding switches.
Yes, this is a huge theme in biochemistry. Both serine and threonine have hydroxyl groups, that same OH handle we saw in tyrosine. That little handle is a perfect target for an enzyme called a kinase.
And a kinase attaches a phosphate group. Phosphorylation, we hear that term a lot.
It's absolutely critical. A phosphate group is big, bulky, and has 2 negative charges. When you attach that to a serine or threonine, you drastically change the local environment. You're adding this big, charged group that can change the shape of the protein.
So it's like an on-off switch.
It is the universal remote control for protein activity. Phosphorylation turns enzymes on, shuts channels off, signals for proteins to move around the cell. And it all happens right there on the serine or threonine side chains.
But the real dramatic character in this group has to be cysteine.
Cysteine is the structural engineer. It has a sulfhydryl group, -SH. Sulfur is a really interesting atom. If you have 2 cysteines near each other and the environment is oxidizing like the environment outside the cell, they can react and form a covalent bond with each other.
This isn't just a weak attraction. This is a real strong bond.
This is a strong covalent cross-link. We call the bonded pair cysteine. Think of it as a molecular safety pin. It locks the protein structure in place. This is crucial for proteins that have to survive in the harsh environment outside the cell, like antibodies or hormones.
Insulin is the classic example in the texts, right?
It's the perfect example. Insulin is actually 2 separate peptide chains that are synthesized, what keeps them from just floating away from each other.
Cysteine safety pins.
Exactly. Disulfide bonds hold the 2 chains together in the precise shape needed to bind to its receptor.
So without cysteine, insulin just falls apart.
And the metabolic regulation of sugar collapses. It's that simple, that direct.
Okay, group 4, the positively charged or basic group. Who do we have here?
Lysine, arginine, and histidine. These all carry a full positive charge at physiological pH, so they are very hydrophilic. But histidine is the one to watch here.
Why is histidine so special?
It has to do with its pKa. The pKa of histidine's side chain is around 6.0. Physiological pH, as we said, is about 7.4. That is close enough that histidine sits right on the fence. It's the only amino acid side chain that can easily flip-flop between being positively charged and being neutral with just a tiny change in the local environment.
It's chemically versatile. It's on the edge.
It's the most versatile actor in the cast. Because it can flip-flop so easily, it is very often found in the active sites of enzymes where it can pass protons back and forth to catalyze reactions. It's a buffer. It's a proton donor. It's a proton acceptor. It does it all.
And finally, group 5, the negatively charged or acidic group.
This one's simple. Aspartate and glutamate. They have carboxyl groups on their side chains, so they carry a negative charge. In the body, they're technically the conjugate bases, which is why we call them aspartate and glutamate rather than aspartic acid and glutamic acid.
And they are key for ionic interactions, right? They can form bonds with the positive guys we just talked about.
Precisely. A positive lysine next to a negative aspartate can form an ionic bond, or salt bridge. It's like a tiny magnet holding parts of the protein together.
Okay, so we've met the lineup. We know who is who. But Harper's biochemistry reminds us that sometimes things aren't what they seem. We have to talk about clinical red herrings and toxic mimics.
This is where biochemistry becomes a detective story in the truest sense. We have the 20 standard amino acids that our DNA codes for. But nature is full of mimics.
And some of them are incredibly toxic.
Highly toxic. Harper spends some time discussing what are called non-protein amino acids. These are molecules that look like amino acids, they act like amino acids, but if you eat them, they can wreak havoc because they don't behave exactly like the standard ones our bodies are built for.
Let's talk about lithyrism. This sounds like a tragedy straight out of a history book.
It is. It's a disease associated with famine. Historically, when people are starving, they sometimes resort to eating a legume from the genus Lathyrus, the grass pea. It's an incredibly hardy plant. It survives drought when wheat and corn die, but it contains a toxic amino acid derivative.
What's this specific molecule?
There are a couple of culprits, but one key player is a compound called BAPN, or beta-aminopropionitrile. It looks enough like a normal amino acid to get involved in the body's chemistry. Specifically, it potently inhibits an enzyme called lysyl oxidase.
And what does lysyl oxidase do?
It's responsible for the final step in building strong collagen and elastin. It forms the covalent cross-links that give your connective tissues their strength. It's what makes your tendons tough and your arteries elastic.
So BAPN stops that cross-linking, the scaffolding fails.
The entire scaffolding system fails. The collagen becomes weak and fragile. The bones deform. The spinal cord gets compressed. People who suffer from lathyrism develop severe skeletal deformities and a spastic paralysis. They lose control of their limbs. It's a permanent, devastating condition caused by a single imposter molecule disrupting the machinery.
That is absolutely terrifying. And there's another mystery mentioned in the books, the Guam mystery.
This is a famous medical puzzle. For decades, among the Chamorro people of Guam, there was an incredibly high rate of a strange neurodegenerative disease. It was like a mix of ALS, Parkinson's, and dementia all rolled into one. For years, nobody knew why.
And the suspect is another one of these amino acid mimics.
Yes, a molecule called BMAA. It's found in cycad seeds, which were a part of the traditional diet, especially after being processed in a certain way. BMAA is a non-protein amino acid. The leading theory is that it looks enough like our neurotransmitters or standard amino acids to confuse the system. It might get incorporated into proteins where it doesn't belong, causing them to misfold, or it might overexcite neurons until they die, a process called excitotoxicity.
It just goes to show how precise our biology has to be. We rely on the standard 20. If you introduce a counterfeit, the results can be catastrophic.
It really emphasizes why the body is so careful about what it keeps. And speaking of keeping, the kidney filtration stat you mentioned in the outline is wild.
Yeah, run that by me again. I found that fascinating.
It's a testament to how valuable amino acids are. Your kidneys filter about 50 grams of free amino acids every single day out of your blood. That's a huge amount of material. But if you check a healthy person's urine, there's almost zero amino acid content.
So we reabsorb it all.
Practically all of it. Over 99%. Amino acids are energetically expensive to make. They're rich in nitrogen, which can be hard to come by. The body does not throw them away. We have these highly specialized transporter proteins in our kidneys to grab them and pull them back into the blood. If you are peeing out amino acids, that's a major sign that something is very wrong.
Okay, let's move from the individual suspects to how they work together. We need to join forces. Let's talk about the peptide bond.
This is the handshake, the connection. To build a protein, you have to link these amino acids together in a chain. We do this through a condensation reaction.
Condensation meaning water is removed.
Right. It's also called a dehydration reaction. You take the carboxyl group of one amino acid and the amino group of its neighbor. You remove a water molecule, an OH from the carboxyl and an H from the amino, and you form a strong covalent carbon-nitrogen bond. That's the peptide bond.
And there is a directionality to this, right? We read it like a sentence, and you can't read it backwards.
Always. There's a strict convention. We read from the N-terminus, which is the end with the free amino group, to the C-terminus, the end with the free carboxyl group. So if you see a sequence written serglytere, you know for a fact that serine is the start, the N-terminus, and tyrosine is the end, the C-terminus.
And pteroglycer would be a completely different molecule.
A completely different molecule with a completely different shape and function. The order is everything.
Now, Leninger gets very specific about the physics of this bond. It's not just a simple rope holding them together. It's described as a rigid plate.
This is one of the most important concepts in all of protein folding. You might think a long chain of single bonds would be super floppy, like a string of beads where every connection can swivel. If that were the case, the protein would be a chaotic mess. You would never be able to find a stable, consistent shape.
But the peptide bond is different.
It is, because of resonance.
Resonance meaning the electrons are shared.
Exactly. The electrons are delocalized. They're shared between the oxygen, the carbon, and the nitrogen of the peptide bond. This gives the bond partial double bond character, and what that means physically is that you cannot rotate around it. It is locked. It is stiff.
So it's flat.
It's planar. Those 6 atoms, the alpha carbon of the first amino acid, the C and O of its carboxyl group, the N and H of the next amino acid's amino group, and the alpha carbon of that second amino acid, all lie in a single, flat, rigid plane.
So the backbone isn't a floppy noodle. It's more like a series of stiff playing cards hinged together at the corners.
That's a perfect analogy. The
Oh.
cards themselves, the peptide bonds, are stiff plates. The only rotation of the backbone happens at the hinges, the alpha carbons.
Yeah.
This limitation is absolutely crucial. Because the chain is semi-rigid, it dramatically limits the number of shapes the protein can fold into. It's what allows for consistent, ordered structures like alpha helices and beta sheets to form.
So the rigidity is a feature, not a bug. It creates order out of chaos.
It creates the constraints that are necessary for life. Without that rigidity, proteins might never find their correct functional shape.
Let's talk about the environment again. We touched on zwitterions, but we need to dive deeper into acid-base dynamics. We have these titration curves in the books.
Titration curves are basically maps of how a molecule changes its charge as you change the pH. And every single amino acid has at least two buffering regions.
One for the carboxyl group and one for the amino group.
Right. The carboxyl group usually has a pKi around 2.3, meaning it gives up its proton at a very low pH. The amino group has a pKi around 9.6, so it holds onto its proton until the pH gets very high.
And in between those 2 points.
In between, there's a special spot. It's the isoelectric point, or Pi. That is the specific pH where the net charge on the molecule is exactly 0. The positive charge in the amino group perfectly balances the negative charge on the carboxyl group. At the Pi, the molecule stops moving in an electric field.
It's perfectly balanced.
Electrically neutral.
But Leninger points out something that I think is really cool and counterintuitive. The values in the textbook, the pK values, those are for free amino acids just floating around in water. But inside a protein, the rules can change dramatically.
Context is everything. This is peer pressure at the molecular level.
Give me an example. How does that work?
Okay, so take aspartate. Freely floating in water, its side chain has a pKa of about 3.7. It desperately wants to give up its proton and be negatively charged at physiological pH.
Right.
But now, imagine you take that aspartate and through protein folding, you bury it deep inside a hydrophobic pocket surrounded by greasy, nonpolar residues that hate charge. Or maybe you stick it right next to another negative charge, like a glutamate.
Well, 2 negatives would repel, so it might not want to be negative anymore.
Exactly. The local environment makes it extremely unfavorable to have a negative charge there. So the aspartate might hold on to its proton much, much longer than it normally would. Its pKa could shift from 3.7 all the way up to 7 or 8.
So a buried aspartate acts completely differently than a free aspartate.
And enzymes exploit this all the time. They create these very specific microenvironments in their active sites to force amino acids to have unusual pKa values, which allows them to act as potent catalysts. The protein is literally manipulating the chemistry of its own parts to get work done.
That's incredible. It's like a tiny self-contained chemistry lab.
It is.
Now let's zoom out from the individual bonds to the whole chain. Primary structure. This is simply the sequence, right? The order of the letters.
It is the linear sequence of amino acids, read from N-terminus to C-terminus, and that sequence is the code that determines everything that follows, the structure, the location, the function. It's the blueprint.
The outline mentions titans and minis.
Proteins vary wildly in size. You have tiny peptides like oxytocin, the love hormone, which is just 9 residues long. And then you have a protein called titin, which is found in your muscles, that is almost 27,000 residues long.
27,000.
It's the largest known protein in the human body. It acts like a giant molecular spring that gives your muscles their passive elasticity. But whether it's 9 or 27,000, the sequence is paramount.
And the classic example of why sequence matters is, of course, sickle cell anemia.
This is the textbook case for a reason. It's so clear. Hemoglobin has hundreds of amino acids in its chains. But in sickle cell disease, just one of them is different. At position number 6 of the beta chain of hemoglobin, a glutamate is replaced by a valine.
So glutamate to valine, that's a group 5, negatively charged, to a group 1 nonpolar greasy one.
Exactly. You've swapped a charged hydrophilic residue that loves water for a sticky hydrophobic one that hates it.
And that position is on the surface of the protein.
It is. Normally, the glutamate would be perfectly happy on the surface, interacting with water. But the valine that replaces it is exposed, and it hates water. It creates this sticky patch. When the hemoglobin loses oxygen and changes shape slightly, that sticky patch on one hemoglobin molecule latches onto a complementary hydrophobic pocket on another.
And they start to clump up.
They start to polymerize. They form these long, rigid fibers that distort the entire red blood cell, forcing it into that characteristic crescent or sickle shape.
All because of one wrong letter in the entire protein sentence.
One letter changes the surface properties. The surface properties change the protein's interactions, the interactions change the cell shape, and the cell shape causes the entire disease pathology. It's a perfect chain of consequence, from the molecular to the clinical.
The sources also talk about using these sequences to trace history, evolutionary tracing.
This is where we get to be molecular historians. Because amino acid sequences are encoded by DNA, and DNA is passed down through generations, we can look at the sequence of a protein in humans and compare it to the sequence of the same protein in chimps or mice or even yeast.
And we look for what are called homologs.
Homologs are proteins that share a common evolutionary ancestor. Within these homologous proteins, we can look for invariant residues. These are the positions in the sequence that never change, no matter what species you are looking at. Cytochrome C is a great example.
Why do they never change?
Because they are absolutely essential for the protein's function. If that residue changes, the protein breaks, the organism dies, and that mutation is not passed on. Evolution preserves them. These are usually the residues in the active site or those critical for folding.
And then you have the other kind, the variable residues.
Right, the variable residues are the parts that change freely over time. They are the parts that don't matter as much to the core function. We can use the rate of change in these variable positions to estimate how long ago two species diverged from a common ancestor. It's a molecular clock.
So our own proteins are living historical documents.
They are. They contain the record of our entire evolutionary past.
Before we wrap up, we have to touch on how we know all this. The methodology. How do you actually read the sequence of a protein?
Well, historically, it was a heroic effort. We have to give all the credit to Frederick Sanger. He was the first person to ever sequence a protein insulin back in the 1950s. It took him years.
And he proved that proteins actually have a fixed, non-random sequence.
That was the key insight. Before Sanger, some people thought proteins were just these random amorphous blobs of amino acids, or maybe variable mixtures. Sanger proved that a protein has a precise, defined chemical formula, just like any other molecule.
And then came Edmond degradation.
Edmond degradation was the workhorse for decades. It's like carefully unstringing a necklace one bead at a time. You use a chemical to label the N-terminal amino acid, you gently pop it off with acid, you identify what it is, and then you repeat the cycle on the new N-terminus.
It sounds incredibly slow.
It was slow, it was painstaking, but it was the gold standard for a very long time.
And today, what do we use now?
Today, it's almost all done with mass spectrometry. We don't peel the beads off one by one anymore. We take the protein and use an enzyme like trypsin, which acts like a molecular pair of scissors and cuts the protein into smaller fragments. Then we put those fragments into the mass spectrometer. It basically weighs them with incredible precision by measuring their mass-to-charge ratio, MZ/MZ. We can even break the fragments apart inside the machine and weigh the pieces. A powerful computer then takes all that mass data and reconstructs the full sequence in seconds. It has completely revolutionized the field of proteomics.
And of course, now we have genomics.
Right. The vast majority of protein sequences we know today are not from sequencing the protein itself, but from reading the DNA sequence in the genome and using the genetic code to predict what the protein sequence will be.
But you still need mass spec.
Oh, absolutely. You need mass spec to confirm that the protein is actually there in the cell, and more importantly, to see how it's been modified, like that phosphorylation we talked about. The genome tells you the blueprint, but mass spec tells you what's actually been built and what it's doing right now.
So we've cracked the case. We've identified the 20 suspects. We know their quirks. The rigid proline, the bridge-building cysteine, the versatile buffering histidine. We know how they link up with those rigid peptide bonds and how their sequence, their primary structure, dictates their 3D fate.
And we know that misidentifying just one like in sickle cell or getting tricked by a toxic mimic from a plant can bring the whole system crashing down.
It really changes how you look at a steak or a soybean or even just your own hand. You aren't just seeing stuff. You're seeing a precise, logic-driven chemical machine built from a very specific cast of molecular characters.
And here's the thought to leave you with something to mull over. We talked about the meteorites. We talked about the 20 standard amino acids that life uses. But why these 20?
Yeah, that's a good question. Nature has hundreds of different amino acids available.
Exactly. Meteorites are full of amino acids we don't use. Plants and bacteria make all sorts of weird amino acids that we don't incorporate into our proteins. So why did life on Earth settle on this specific alphabet? Was it just an accident? A frozen moment of chance? Billions of years ago when the first cell emerged?
Or is there something special about them?
Or is there a deeper chemical logic, a kind of chemical destiny, that makes these 20 the only 20, or the best 20, that could be used to build a complex, adaptable, and evolvable life form.
That is the ultimate mystery, the origin story.
It is. We can build rhinoceros horns and spider webs and the enzymes that digest your lunch, all from the same 20 letters. It's a limited alphabet, but it produces an infinite poetry.
I love that. A limited alphabet, but an infinite poetry. Next time you hear the word protein, don't just think of a nutrition label. Think of that poetry. Thanks for diving deep with us.
A pleasure as always.